What we're doing is this 5-week class is really in the field of what we call synthetic genomics. So I'm sure everybody's familiar with the Human Genome Project, right? Completed around 2001. And the huge deal with the Human Genome Project is for the first time we could really read DNA sequences of the entire human genome. And since that Human Genome Project, our ability to sequence, to read DNA has increased tremendously, exponentially. We can now sequence a human genome for about $2,500 in about 2 weeks. Okay, so it's really become almost trivial to get the sequence of DNA that you're interested in. So following the ability to read DNA, now we've got this new technology, the ability to actually write DNA. To, rather than starting with DNA that's pre-existing as it is in nature, to really be able to create DNA of any sequence that we want. And that's really the field of synthetic genomics. It's trying to synthesize and create DNA, either replicating what already exists, so simply recreating what's there, which is what we're going to be doing, or starting to write and create new DNA sequences. So many of you have probably heard about kind of this idea of synthetic life, creating life in the laboratory, pioneered really by Craig Venter in about 2010. They made the first bacterial cell, where that cell was able to live off of DNA that was completely made in the lab. So you can synthesize DNA in the lab, you can put it into a cell, and that DNA actually works. It can actually support the entire life cycle of an organism. And if you were here for John Glass's talk a couple weeks ago, he was one of the leaders on that team, and he talked about making that bacterial genome. Now, the genome that they made was from Mycoplasma, which is a very simple bacteria, and they pretty much just recreated the naturally existing bacterial genome. Not too many changes. Because again, when you're writing DNA, you don't want to make so many changes that you're going to kill the organism, right? That DNA still has to be able to code for life and code for function. So you have to be kind of cautious when you're making changes. So for that first project, they were very cautious and made very few changes. A couple months ago, back in March, April, a paper came out, the first synthetic eukaryote, not a full genome because eukaryotes, remember, are much bigger, but one chromosome of a eukaryote, and that was in yeast. And that chromosome used the pre-existing sequence, but it made much more substantial changes. So in fact, what we did was we took out almost a third of the DNA from that chromosome. So we made it kind of a smaller, more compacted chromosome. And that chromosome actually worked just fine. So you can actually make pretty radical changes to the DNA, to the chromosomes, to the genome, and the organisms can still live with it. And obviously this is a really important tool for learning about, what's in DNA, what's in a genome, besides the genes that are important for that genome to function. So we'll talk about those two projects, the synthetic bacteria and the synthetic yeast, probably in session 2 and session 3. We'll talk about them a little bit more in depth in terms of how you do a whole genome, what kind of changes might you want to make, what kind of changes can you make. But for right now, we'll get back to kind of the simple idea of synthetic genomics. We're not going to be able to make a full genome. The synthetic bacteria took about 15 years to make. The synthetic yeast chromosome took about 7 years to make. We just don't have that kind of time. So what we're going to do, instead of building a genome, so the class I teach at Loyola is build a genome, right? We're building that synthetic yeast piece by piece. What we'll do over the next 5 sessions is building a gene. So one small segment. But all the techniques that we're using are exactly the same techniques that you use to build a genome, just on a smaller scale. So what we want to do is, the gene that I picked is green fluorescent protein, GFP. It makes cells glow green. So if we're successful, we'll have taken DNA, and at the end, we'll have introduced it into cells and gotten our cells to glow green. Okay? Now, what's actually going to function and make the cells green is the GFP protein, right? Protein is what actually functions in cells, in this case to make them glow. That protein, however, comes from DNA. The information for the protein is encoded in the DNA and it gets converted into protein. You guys remember, this is what we call the central dogma of biology, that DNA gets converted into RNA, and then it gets converted into protein. So we'll be introducing DNA into cells and hoping that those cells produce protein and glow green. Now, in order for DNA to get converted into protein, we need a couple things. One is what we call the coding sequence. In other words, the sequence of the protein. What do we want to make, right? What amino acids are going to encode this protein to make the cell turn green? But we need a couple more things in order for the cell to recognize this DNA and convert it first into RNA and then into protein. Does anybody remember how you go from DNA to RNA? What's the sequence that the cell is looking for to tell it where a gene is to convert it into RNA? So the genome is huge, right? There's 25,000 genes. Not all of them are making protein all at once. It's a very regulated process. And the cell is looking for a specific signal in front of a gene to tell it to make RNA. Do you remember what that's called? Close. That does this part, RNA to protein. Promoter. Promoter, exactly. So the promoter is a sequence that comes before the coding sequence and that tells the cell this sequence should be made into RNA so that it can later be made into protein. Any questions about that? Okay, so when we make GFP, it's going to have both the coding sequence and it's going to have a promoter so that it can be converted to RNA and then to protein. Okay? We're going to be making two different constructs, or two different versions of RFP. One is going to have a promoter that just says, make this gene, make GFP. Anytime, all times, and in all places. Just make GFP. And so our cells will turn green. The second is we're going to use a promoter from a different gene. This is a yeast gene called RNR3. That promoter doesn't make RNA at all times. It only makes it under specific conditions, and specifically under conditions of DNA damage. So you know that, like, if you go outside and you get too much UV, or if you're exposed to some kind of a toxic chemical, your DNA can get damaged. And when that happens, your cell responds by turning on a whole bunch of repair factors, one of which is RNR3. Okay? So this promoter only leads to RNA production when DNA has been damaged. Okay? So when we make, first we'll make normal GFP. It's made at all times and in all places with a normal promoter. Then we'll also make RNR3 GFP. So this is going to make green fluorescent protein, but it's only going to make that fluorescent protein when the cell has been damaged. Okay? In our case, we'll probably use UV light. Okay? So our cells normally will look like normal cells. You know, they'll be colorless or white. But once the cell gets damaged, hit with UV rays, they're going to start to glow green. So this should function as a DNA damage sensor. Right? You should be able to tell that your cells have been exposed to UV because they'll turn green. So obviously there's practical implications for where this could be useful. Okay? So this is just to kind of show us the process, the steps of synthetic genomics. This is to actually use synthetic genomics to build something that might have a practical or a working function. Okay, any questions? Oh God, all my scribble's gonna wind up on the internet somewhere. Any questions?
You'll be immortal.
actually going to make this? All right, the first question is, well, how are we going to get the DNA into cells? Now, most DNA in the cells is on chromosomes, right? I just told you we don't want to build a chromosome because that's huge. That'll take us years to build. So instead of putting in the DNA on a chromosome, we're going to put in just our gene, our GFP coding sequence, and our promoter, either the normal one or RFP. Our promoter and our coding sequence, that's all that we really want in the cell. So instead of having the rest of a chromosome around it, what we're going to do is use a plasmid. Anybody know what a plasmid is? Right. So bacteria and actually yeast too naturally carry, in addition to, here would be, their big old chromosome or chromosomes that would be huge, they also just naturally happen to carry these small little circles of DNA that are called plasmids. And plasmids are awesome because they can come in and out of a cell. The cell doesn't need them to live. You can pull them out and put them back in. And plasmids are perfectly happy, if this is the natural plasmid, to have us insert foreign DNA into that plasmid, and then we can put it back into the cell, and the cell will actually maintain this plasmid with the foreign DNA, and assuming you've got all the right signals, in other words, the promoter, it will express this foreign DNA, convert it to RNA, and eventually convert it to protein. Any questions about that? So what we're going to be making, what we're going to be combining, such as the promoter, the GFP, and this plasmid, and then introducing that into cells. Okay, so where's all this going to come from, right? We need our 3 different pieces. The plasmid is going to be a naturally occurring plasmid that comes from bacteria, and that's one that I'm going to give you. Okay? So this originally was isolated from bacteria. We can now manipulate it in the lab. The promoter, I told you, is a piece of yeast. It's normally a yeast gene, so it's a small piece of the yeast chromosome. And then there's the GFP coding sequence, and that's what we're going to be making today.
And the first one, that's not yeast, the promoter.
This promoter?
Yeah.
Well,
Are they both?
So the RNR3 promoter is a yeast promoter. When we just make GFP by itself, we'll use its natural promoter. So that doesn't actually come from yeast. Anybody know what GFP is originally made or produced? Jellyfish. Yeah, deep sea jellyfish tend to fluoresce under color. So the original GFP has been isolated from jellyfish. There's now a whole rainbow of fluorescent protein colors. The red fluorescent protein, which we'll probably use as a control, isolated from coral that fluoresce too. So if you go down and you find all these cool little marine organisms and you sequence the DNA, and you find all these cool genes that then you can then manipulate them.
So if there's a promoter to start it, what stops it?
Okay, so there does need to be, at the end, what we call a transcription terminator sequence, which is going to be, for our purposes, as we include as part of the coding sequence.
Okay.
Yeah. Okay. Other questions? Okay, so where are we going to get the GFP coding sequence from? We could go to jellyfish DNA and amplify that GFP coding sequence and use it. But instead what we're going to do is we're going to make this DNA from scratch using synthetic DNA. So what that means is that we're not going to start from any kind of natural or pre-existing template. And this is where synthetic genomics, why we call it synthetic DNA, is different, is that it doesn't start with any pre-existing template. And that really frees us up, right? We're not limited by any kind of naturally occurring sequence or genes. If we're making DNA from scratch, the great thing about synthetic genomics is I can make any sequence that I want. All right, so the GFP coding sequence is about 750 nucleotides of DNA long. So how am I going to make this from scratch? What do I mean by from scratch? Well, this is where our friends, the organic chemists, have really helped with this field, because organic chemists can essentially do DNA synthesis, or something very similar to DNA synthesis, by doing organic chemistry. Okay? So they can put together nucleotides into a chain of nucleotides. Now that process of putting together a chain of nucleotides is incredibly accurate. It's about 99.5% accurate. If I tell the organics chemists I want 3 nucleotides, an A nucleotide, then a T, then a C, they'll put it together. Each linkage from the A to the T, from the T to the C, will be about 99.5% accurate, which if you talk to an organic chemist is awesome for chemistry. Okay, that is a fabulous rate for chemical synthesis. However, for biological synthesis, that's not really great, right? Biology, we need a perfect stretch of nucleotides without any errors. And errors are what we call mutations. Mutations are bad. Mutations are not what you want. So if I ask an organic chemist to put together 3 nucleotides for me, an A, a T, and a C, the accuracy will be 0.995 for the first linkage and 0.995 for the second linkage. And I didn't do the math, but it winds up being very good, greater than 99% accurate. So if I ask an organic chemist to make me a small piece of DNA, they can do that very accurately. Now what if I ask an organic chemist to make me a piece of DNA that's 750 nucleotides long? That's much harder, right? So the frequency of that would be 0.995 raised to the 750, right? 0.995 multiplied out, and that, I did do the math, was 2.7%, 0.027. Okay? So the accuracy of making a long stretch of DNA is not great. So if we want a 750 nucleotide length of DNA, we don't make it all in one big piece because that's not very accurate. Okay? What we do is we break it into pieces and we put together the pieces. So generally our pieces are about 65 nucleotides long. That is, 0.74, so 74%. So the longer a piece of DNA you get, the less likely it is to be accurate. So you just kind of have to find that sweet spot. We could do a 50 nucleotide piece and it would be more accurate. We could do an 80 nucleotide piece and it would be less accurate. 60 is kind of the point, at least for the synthetic yeast project that we've settled on, and most people are in that area. Okay? So what we're going to be doing is we're going to be taking 60 nucleotide pieces of DNA that have been made by a company. You can just go online and you go to the company and you put in your letters, A, T, C, G, G, C, A, whatever it is. of your pieces of DNA, and we're going to take those pieces of DNA and we're going to stitch them together to make the full 750 nucleotide piece. Any questions about that? So if our goal is to produce 750 nucleotides of GFP, we get, I think there were 20 of these. 20 pieces, all about 60 nucleotides long. And we combine them all together. These are going to be called your assembly oligos. So DNA is made of nucleotides. Several nucleotides together are called oligonucleotides. Because we're lazy, we call this oligos. So oligo is an oligonucleotide, which is a small length of DNA. Any questions? Okay. Anybody notice a problem with my math? We want a piece that's 750 nucleotides long. We have 20 pieces that are each about 60. If you do the math, that's 1,200 nucleotides. That's longer than our gene, right? Right? The reason for that is because some portions of this gene have been synthesized twice. If we had our oligos, 1, 2, 3, 4, 5, 6, 7, all 20 of them, right? Dot, dot, dot. The oligos don't know which order you want them to be in. If we take 20 oligos and mix them in a tube, they don't know that they're supposed to line up in the correct order. Okay? The way we force them to line up in the correct order is we take advantage of the fact that DNA is double-stranded, right? You guys all remember the base pair rules of DNA, right? That for any piece of DNA, if here's one strand of DNA, DNA is always double-stranded. A always pairs with T, G with C, A and T, G and C, G and C, A and T. Okay? So they always base pair together like that. Okay? So if we have one sequence here, let's say this was A, C, T, G, C, A, this portion here, if oligo 2 has what we call the complementary sequence down here, T, G, A, C, G, T, these two oligos are going to base pair together, because A's and T's and G's and C's always base pair. They're going to base pair together, and that's going to put oligo 1 and oligo 2 together in the correct order. Okay? So they're essentially going to stick together by hydrogen bond newer base pairing. Okay? So each of our oligos, 2 overlaps 3, 3 overlaps 4, 4 overlaps 5. Okay? So there's a unique sequence at each of these overlaps that causes the oligos to stick together in the right order. So that's why we've got so much extra DNA, because these overlaps, that length of DNA has been synthesized twice. Once on oligo 1 and once on oligo 2.
Does that help also with the statistics of accuracy? Like, I'm just thinking, you know, using your algorithm of 95% or 99.5%. If you've got an overlapping.
If you've got an overlap, if one's got a mistake here, they'll still base pair together, but the chances are that if one has a mistake, the other's overlap is probably good at that position. So yes, there's less likely for your final product to have a mistake in it. Because if one is bad, the other one's probably good.
And how many nucleotides of overlap are there?
So each of these overlaps is designed to be about, I want to say 60 nucleotides long. And we'll go through, once we set up our reactions now and put them in, we'll go through how, if you're starting with the GFP gene, how do I do this? How do I know what my oligos should be? How do I break up a gene into pieces to know what to order? Okay? This is actually a very complex algorithm to make sure that the overlaps are correct.
And the overlapping regions code for the protein as well?
Yes. Yes.
Isn't they, each oligo is 60 nucleotides long, or the overlap is 60 nucleotides long?
Yeah, that's why I just said to myself that both don't seem to be right, do they? Yeah, you can't have both at the same time.
You wouldn't have anything to connect to the next piece.
Let's look at the program.
20 to 15?
Yeah, that's when we do the program. I think I have my overlaps wrong.
And are,
Because I know you can set your oligo length to what you want, you can set your overlap length to what you want. So for 60 nucleotide oligos, I think you're right that this length of overlap is too long. I've seen anywhere from 20 to 60.
and then what about this space in between? Does it have to overlap so that the top two pieces, like 1 and 3, are right
Bump
next to
into
each
each
other?
other?
Yeah.
Okay, so that's what we call whether your oligos are gapped or ungapped.
Okay.
There's advantages to each. If your oligos are gapped, that means 1 and 3 don't touch each other. Okay? So what we're going to do now, the process we're going to do is we're going to take your assembly oligos, which are all 20 oligos mixed together. We're going to put them in a tube with nucleotides, and we're going to add an enzyme into it. Okay? And what the enzyme's going to do is any place where it's single-stranded, the enzyme's going to fill in and make it all double-stranded. Okay? So the enzyme is going to turn this partially overlapping 20 nucleotides into one smooth piece of double-stranded DNA. And the reason for that is because the oligos that we have are gapped. They do have these open regions between them. Now the advantage for putting in those gaps is I told you chemical synthesis is 99.5% accurate. Biological synthesis by the enzyme is 10 to the minus 6. So error rate's 1 in a million. Yes, 0.000012341. Okay? Much better. Or I guess, sorry, I did it the wrong way. If the accuracy of chemical synthesis is 0.995, the accuracy of biological synthesis, 1, 2, 3, 4, that's 1 out of a million errors, right? I got that right? Okay, so biological synthesis is much more accurate than chemical synthesis. So the advantage of using the enzyme to fill in is that it's going to be much more accurate. Okay? The advantage of doing your oligos so that they essentially collide with each other and don't make gaps is that they stick together much better, right? The longer the overlap is, the more likely that these two oligos are going to find each other and bind to each other properly and in the right order. So again, that's why we have got to find that sweet spot where you have the accuracy of making the nucleotides, but you also have the accuracy of the oligos binding to each other correctly. So that's one of the reasons why the overlap can vary. Longer overlaps mean that they're more likely to come together in the right orientation. Shorter overlaps, though, mean that you have more biological synthesis and more accuracy of actually making the DNA.
The shorter overlap increases the chance to bind correctly? Seems like
No,
that's-
the longer overlap.
Okay.
The shorter overlap is advantageous because you've got more biological synthesis and more accurate synthesis.
Okay,
Yeah.
but you make more accurate stuff.
You make more accurate stuff. So it's always a trade-off, and for each project that you're designing, you might decide, which one you prefer. And you might have, if you find that you are trying to assemble a gene and it doesn't assemble, one of the things you might do is increase that overlap length to make it more likely that your oligos go together properly. Any questions? Again, we'll come back while our reactions are in there and kind of talk about this process of breaking up your gene into oligos. But let's go ahead, because this actual process of synthesis of the enzyme filling gaps is going to take a good hour and a half once we put it in.
So it's like a good way to look at that. You basically want it to be as short of an overlap as possible while still getting good assembly.
Yes.
Okay.
Yeah.
Just make sure.
Yeah.
What's the enzyme that does the oligo assembly?
Okay, so the enzyme is going to be a variety of TAP, which some of you know from PCR. It's the same enzyme that we use in PCR. We actually use, there are now a lot of varieties of TAP which vary in their efficiency and their accuracy. So we actually use one that's got a high accuracy and a high efficiency that's called Hercules. Strong, because you need a strong enzyme to do this. So a better enzyme is going to be more accurate and more efficient in putting together the oligos. Okay, so what you have on your instruction sheets, we call this template-less PCR because we don't have a template, right? We're not amplifying a pre-existing gene. We're putting it together from oligos without any kind of pre-existing template. So in the template-less PCR, one of the things you'll notice is that everything you're going to be working with today is on ice. The enzyme, the Hercules, is temperature sensitive. Nucleotides are also temperature sensitive. So you want to leave everything on ice as much as possible. Never like hold a tube. I can pick up one that won't matter, but never hold a tube like that in your hand because your hand's at 37 degrees and you're warming up, you're denaturing the nucleotides and the proteins. So, you know, flip the top open and then leave it on ice as much as possible. Okay, so what you'll see down in the procedure is first thing we're going to do is assemble the GFP gene. So to do that, you need master mix, which essentially is the enzyme Hercules plus the buffer, which puts it at the right salt and the right pH. So that's MM, that's your enzyme. Then you have nucleotides, okay, which is what you need to actually make the DNA. You have your assembly oligos, which is that mix of 20 oligos that are going to make the GFP gene. And you have the DNA template. In this case, we don't have a DNA template, right? So you're just going to use water. Okay? So it's oligos, nucleotides, and enzyme, essentially. Okay? Now, every time we do a reaction, let's say your reaction didn't work. You got nothing amplified. You wouldn't know if the problem was, you know, your design, that there's something wrong with your oligos and how you designed it, or if you just messed up. Like, you know, we've all done it and you forgot to add an enzyme or something. You know, you forgot to add something into the mix. So every time we run a reaction, we try to run what's called positive control, which is something that you've done before that you know should work and should give you a band, should make DNA. So that's going to be the second one, is your positive control reaction. So you're using the same master mix, you're using nucleotides, and now you're using control oligos, which should also be on there, and you're using a template. So this is PC, or positive control template. Okay? So essentially all this reaction is I'm giving you a small amount of DNA, and you're going to use the oligos to amplify or make more of that DNA. And this is something that I've done before many times. I know it should work, assuming you've added everything correctly. Okay? The other reaction we always do is what we call a negative control. So let's say, you know, somebody before you has been working with the pipette. It's completely coated with all kinds of DNA from their previous projects, and you run it on a gel and you see DNA, you think it's the right thing, but in fact it's not. It's just a contaminant. Okay? We always want to be on the lookout for those. So every time we also run a negative control, this is something that shouldn't give you any DNA. Okay? It should show no amplification, no DNA. And that confirms for you that you don't have like a massive amount of DNA on the bench, on the pipette, on your hand that's contaminating your reactions. So you'll see all that's in there is the master mix, which is the enzyme, nucleotide, oligos. These are not assembly oligos, though. They should not go together. These are oligos that should not produce a gene, and a little bit of water. Okay? So that one should be a blank lane when we run it on the gel. Okay? Any questions on that?
How do they control all of this in the assembly line?
Okay, so the control oligos are, instead of coming together to make a gene, what they're doing is they're amplifying a pre-existing gene, and that's a much more efficient process. So they should be much more likely to give you a band on the gel than the assembly oligos. The assembly oligos, it's a very inefficient process, right? Because all 20 oligos have to come together. This one's only 2 oligos, so much more likely to work. Other questions? Good? Okay, so to do this process of putting together the oligos to make a gene, we repeat the process of assembly we're going to do it about 30 different times. So we call this cycling, or repeating the process over and over and over again to increase the amount that we get. We do this cycling by adjusting the temperature. We raise the temperature so that the oligos separate from each other. We lower the temperature so that the oligos anneal together. Okay? So to facilitate these temperature transitions, we don't use the large tubes, which is what your reagents are all going to come on. We want to use these very small tubes, which are very thin walls. They conduct the heat really well. So we actually set up the reactions in these little tiny tubes because they conduct the heat better and allow the oligos to anneal. Okay? So you're going to take 3 of these little tubes. You can label them 1, 2, and 3 for your assembly, your positive control, and your negative. I always like these guys because they hold the tubes really well. So 3 tubes, and then you can simply add all of your reagents into tube 1, into tube 2, into tube 3. Any questions about that? Hopefully they're all well labeled. There's enough on each ice bucket for a pair of people to share the reagents. Good with that? Okay, everybody good with pipetting? Do we want a quick review of pipetting? Yes. Okay. So you're going to have 3 different pipettes. Hopefully you can tell what size they are by the top. They should be called, we might have some variations here, but they should be called the P1000. You use that for volumes that are about 100 microliters to 1,000 microliters. You should have a P200. You can use that for volumes that are 2 microliters to 200. And you should have a P20. You can use that for 2 microliters to 20 microliters. So the number here represents the maximum volume that you can use with that pipette. So for most things, like if we're pipetting either 5 or 10, you can use the P20, or if you want the P200. But generally the P20 is better. Okay?
The P10s, do you not want to use a pipette with a maximum?
No, it's fine with max, yeah. Oh yeah, we do have P10. So P10 is the same thing, it goes from 0 to 10. So I'd recommend using the P20 or the P10, either one. Okay? What you'll see is you've got 3 numbers in the window, and I always kind of count down from the max. So if I'm on a P20, 200 represents 20.0. So you'll see again, they all get the master mix, which is the enzyme in the buffer. They all get more nucleotides. This time, you're either going to be using the control oligos or the amplification oligos. And again, those amplification oligos are 1 and 20. That's what's in that mix. And then this time, for the template, last time we didn't have a template, we just had water. This time, your template, I called it the GFP template. What it actually is your templateless PCR reaction, right? So you're taking a little bit of your templateless PCR reaction and putting it into the finished PCR. Okay. You'll notice that in step 5, you're going to be taking your, it says take your liquid from your tube 1 of your templateless PCR, your assembly PCR, put it in a new tube. To that tube, add 180 of water. So what we're really doing is we're diluting your templateless PCR. So you're taking that reaction, you're adding water, and you're only taking 5 microliters of it. So, you know, your whole reaction was 25 microliters, you're adding 180, so you should have 205 microliters of diluted templateless PCR. You're only using 5 microliters in your finished PCR reaction. Okay, so a very little amount. Any questions about kind of finished PCR, what it's about?
So for the negative control, we're not even going to use our PCR, we're just going to use 5 microliters of water.
Yes.
Okay.
So what we're doing now, your reactions that are in now, when they're done, your positive control, we're going to save it and run in our gel next week. So you're going to transfer it into a larger tube that you label, I think I said TPCR for templateless PCR PC, positive control. Your negative control you're going to transfer into a new tube, save it for next week, label it TPCR NC. So those are both reporting on this reaction. Did you set up your templateless PCRs correctly? When you set up your finished PCRs, you're setting up a whole new set of controls. So a new positive and a new negative. Does that make sense? Because you want a reporter, did you set up your templateless PCR correctly? You want a separate reporter, did you set up your finished PCR correctly? Does that make sense? And then your amplification, again, is the master mix, the nucleotides, the oligos, and then we have to wait for our templates to be done. But you can essentially, and I would recommend, since it takes a little while to set things up, is to set them up now. Your positive and your negative controls you can set up, and you can set up everything for the amplification except for the GFP template, which is obviously cooking along right now.
Do you get the first round controls carried into the second round, so you end up with like 6 tubes, or do you end up with 3 tubes at the end of the second?
You should wind up with 6 tubes because your TPCR positive and negative should be separate. You don't reuse them. You make a brand new positive and negative for this set of reactions. Other questions? I like every time I'm setting up to set up a fresh positive and negative because you could have set everything up right the first time and messed up the second time or vice versa.
But do you get enough amplification in the assembly round of your, say, your like positive control?
Well, the positive control isn't an assembly reaction for both of them. Your positive control is a traditional PCR.
Oh.
So yes, you
Okay.
get enough. Yeah.
And do we have a different batch of oligos that just have?
Yes, you have a new batch of everything. So if you want, what you can do is to take everything that was on your ice and toss it into the waste. And then I've got a new set of everything that I can give you, nucleotides and master mix, that way you know you have enough. Maybe for us, which that's the address, but if you simply Google open wetware, build a gene 2014, you'll get there. And I put up a little description about what we're doing, and I put up links to each of the 5 sessions that we're going to be doing. So again, today we did gene synthesis. In 2 weeks, what we'll do is we'll do gel electrophoresis. That's a way for us to actually look at the DNA we just hopefully made today and double check that it's the right size. And then we'll purify the GFP gene that we made today. We'll get rid of all the old buffer, the enzyme that we used to make the gene so that it's ready to assemble. The third session will actually assemble the GFP gene with that plasmid and with the promoter. And we're actually going to do that in 2 ways. This is new, I'm really excited about it. We'll do both Gibson assembly, which is kind of an enzymatic reaction in a tube, again, developed by Dan Gibson of the Venter Institute for their synthetic bacteria project. So this is really pretty cutting edge. And we'll also be doing it a second way, which is to actually use yeast cells to assemble the 3 pieces for us. So all genome synthesis projects, when you're assembling small pieces, you can do it enzymatically in a tube. When you're putting together large pieces of DNA or many pieces of DNA, all genome synthesis projects use yeast. So I'll actually be showing you both versions, the enzymatic in the tube and the yeast version. On week 4, we'll actually take that plasmid plus GFP plus the promoter that we made in yeast. We'll pull it back out of yeast, and here's where we'll put it in bacteria. I told you we need to put it in bacteria because the cells each pick up one piece of DNA. So it allows us to separate the good ones from the bad ones. And then on week 5, this is kind of our ultimate seeing whether or not we actually were able to make what we wanted. So we'll take those bacterial cells and we'll actually screen them, and we'll see which ones are perfect, have exactly what we want, and which ones don't. So hopefully at the end we'll have our complete gene either made by enzymatic assembly or made by yeast assembly. We'll have to see which one works better. So that's kind of the overview, and again, you can click, any of them should have links to kind of the protocols if you want an overview. Subject to change with limited notice. But if you click on session 1, for gene synthesis for oligos, again, you'll get the templateless PCR, you'll get the finished PCR, and then down at the bottom is how you actually design the oligos. Okay? So the way we design it, you got to start with what you want. So this is the sequence of the GFP gene. Again, we're doing the version of GFP called Emerald. So this is Emerald GFP. And this is about 750 bases long. And so the first thing you got to do is you got to copy this. And you take it over to a program called Gene Design, which is going to design the gene for you. And you can see there's many ways you can either design a full gene or you can do all kinds of different manipulations to the gene. And I can talk to you about that if you're interested in some of them. First thing that we're going to do, though, is we're actually going to take that GFP gene and break it into those oligonucleotides. So, you know, if I have my gene, how do I actually know what to order from the chemical synthesis company? And the program will do it all for you. So what you want to design is actually called a building block in this lingo. Different genome projects call it different things. We call it building blocks. So you want to go to building block design with a constant length overlap. You just click on that. And simply input your sequence here. That's not my whole sequence. Okay. Give it a name if you want.
Can we separate the projector and the laptop a little bit so we can see?
You mean can I not stand right in front of the projector? What's your problem?
Yeah, Ryan ought to work for the State Department. He's so diplomatic.
So what the program will do, first of all, a building block, a unit that you want to put together, 750 is really on the large side. Let's say I put in 3,000 base pairs. It would tell me to not put it together all as one reaction, maybe 60 oligos. It would tell me that's too long, break it into pieces. So the first thing that it'll do is decide whether it should be built as one big building block or multiple building blocks. Since ours is 750, it should keep it as one unit. But again, if you put in a huge sequence, it would break it up into smaller pieces. Okay? Then it's going to look at the oligos. And again, here's where you have some parameters you can change. How long you want the overlaps to be. So somebody asked, so I guess the default is 40. So there's 60 nucleotide oligos, 40 nucleotides overlapping between them. Again, you can generate them as gapped oligos where the enzyme's going to fill it in for you. Or if you unclip that box, there won't be any gaps. So the oligos will sit perfectly next to each other without any gaps in the middle. Again, the average length of your building block, again, 740 is about the default. You could adjust it if you really wanted to make a building block that was 1,000 nucleotides, all as one big piece. It would let you do that. The target oligo length, so again, each oligo is going to be about 60, but you can adjust it. You can set the maximum if you want. And then the overlap. TM. Okay, so this is really important. So we went through, you got those 3 steps of denaturing, annealing, and extending. What you really want is at that annealing step, you want each of your oligo pairs to have an equal likelihood of coming together, right? Now some of you might remember that we've got 2 different types of base pairs. We've got AT base pairs, and we've got GC, right? Our oligos are going to base pair together. 8 and T's are joined by 2 hydrogen bonds, and they tend to be weaker interactions. G's and C's are joined by 3 hydrogen bonds, and they tend to be stronger interactions. So if I had an overlap that was 40 nucleotides long and it was all G's and C's, that would be very strong, and those oligos would be very likely to come together. If I had another pair of oligos that overlapped by 40, but this time they were all A's and T's, that would be much weaker, they would be much less likely to come together. So if I had, let's say, oligos 1, 2, 3, and 4, if this one were all G's and C's, it would come together very well. If this one were all A's and T's, it would come together much less well. Okay? What we want to do is we want each of these overlaps to have an equal chance of coming together. So that means instead of each one being a perfect 40 nucleotide overlap, what the program does is it actually adjusts the overlaps for you. So if your sequence has a lot of G's and C's, it's going to shorten the overlap region. If your sequence has a lot of A's and T's, it's going to lengthen it. So that 40 nucleotide overlap is going to be an average. Some will be lengthened, some will be shortened, with the goal of what we call the melting temperature. The temperature at which the oligos anneal should be equal for each overlap region. And this is why we use, I mean, if you were just designing, breaking your 750 into 60 nucleotides and 40 nucleotide overlaps, you could design it by hand really easy, right? But this is what's a little bit harder, and it's why we use the program and the algorithm to adjust them all simultaneously.
I'm missing a little. I'm missing something somewhere, because if we have, if our lengths are 60 and we're overlapping by 40, to me it seems like you couldn't overlap.
20 in the middle.
Put 20 in the middle.
Yeah.
Oh, so that's just overlap on one side? 'Cause it seems,
I see what you're
yeah,
asking.
'cause it seems like you could
Yeah.
overlap by more than half of your
Right.
length.
Yeah. So
It's full.
the idea, the question.
So your whole nucleus one is 60, and then this region of overlap here should be
22. For 740, right?
So if each one's
if that
60,
was
you can't
40.
overlap by 40 on both sides.
Yeah,
Right.
yeah.
Especially not if
Let's
you're going to end up
see
with
what
a
it
gap.
gives us.
Like you'd have to
Maybe
do
it's
20
20
and
on
20, then
either
you get a
side.
gap of 20 in the middle.
Okay, yeah, that's what it is. 1, 2, 3, 4.
We have this computer and we're sitting there.
That's 20.
There we go.
Got it.
Okay, so what you get when you hit enter is here you get 4 strands. The top is the desired strand, your 750 nucleotides of GFP. This is the bottom strand, and in the middle are your oligos. And if we go across, what you should see is 20 overlapping oligos. And if you do like I just did and you actually counted it, so this would be oligo 1, should be 60 nucleotides long, oligo 2, 60, oligo 3, 60, and if you count them like I just did, it's 60 nucleotides long, 20, 20,
So you're
20,
specifying
ballpark.
the
So-
total. Sorry.
It actually says above their average oligo length is
59 base pairs instead of 60. So it's going to lengthen some and shorten some. in order to adjust those overlaps.
So when you take that and send it to them,
So what you do is you click on FASTA format, which is just the standard format for DNA. You click Assembly Oligos, and what it gives you here is a list of, in this case it made 18 oligos. You can see they average 60 base pairs, but some of them have been lengthened to increase the overlap, some of them have been shortened to decrease the overlaps.
And these are all in order, so if you were to pick this apart, you could connect it at one end and reconnect it.
Yes, absolutely. Yep. Other questions? So these would be, this is what was in your assembly mix, and then the first and the last would be the ones that you use for finished PCR. Any questions on that? Okay, now, so you could use this just to input the GFP gene straight out of jellyfish with the thought that you're going to make the jellyfish GFP gene, you're going to put it in bacteria, the bacteria should express that GFP gene, everything will be fine. It's actually, I always find it's really amazing that biology works that way, that you can put something from jellyfish into bacteria and, you know, it works and it's great. But it's not perfect. So one thing that we have to deal with is a phenomenon that we call codon bias. So if you remember back, there are, when the DNA gets made into protein, it gets read in groups of 3, which are what we call codons. So if we had AAA, TCA, this would be the first codon, this would be the second. There are 64 different codons.
Thanks.
But there are only 20 amino acids. Right?
Oh, don't hand him that. Ask him what it costs.
Are the units for DNA, the amino acids are what makes proteins, right? So there's 20 different units of protein, 64 different units of amino acid. And the reason for that is that some amino acids are encoded by different codons. So for example, this 3 letters AAA encodes lysine, okay? But the 3 letters AAG and AAT and AAC all encode lysine. Okay? So if your DNA sequence said AAA, it would get made into the lysine protein. If it said AAT, it would also get translated into the lysine protein. Now, I told you it's pretty amazing that it can move genes between species and it works, but the different species aren't exactly all identical. And this idea of codon bias has to do with different species prefer certain codons to others. They can use them all, but they'll prefer certain ones to others. So if you look at all the lysines in all the proteins in a particular species, you might expect kind of, at first thought, well, this one, 25% of them should be coded by AAA, 25% of them should be coded by AAG, 25 and 25, right? Each equally. What you'll find is that that's not exactly true at all. For certain species, they might prefer AAA and use that particular codon to code for lysine more often. And they might use this one less frequently.
Does that have an implication for engineering?
What do you mean engineering? Well, it probably has to do with how fast and how efficiently the protein is translated.
Oh, okay.
Okay? So we call these preferred codons. So for example, I'm just going to make this up. Let's say E. coli might prefer AAA, which means when it sees an AAA, it prefers that codon, it translates or converts it into protein very quickly.
Will you find non-preferred codons as well?
Yes, you will, and sometimes an organism will use a non-preferred codon intentionally, often because it wants to slow down translation. So there are both. Okay? Generally speaking, preferred codons get translated faster and more efficiently, with exceptions, but generally speaking, that's true.
Sorry if I'm getting like too off my tangent, but like how like ballpark like is like what kind of a swing in efficiency can you see between two codons?
So I know for like alanine, I know I've looked at it between yeast and human, and it goes anywhere from, a particular codon might go from 17 to 45% because of the frequency of it getting used
That's significant.
instead of 25. Yeah. So even though you can move a gene, a DNA sequence, between different species, if you move a gene into E. coli that doesn't have the preferred codons, it might not get translated very quickly. Okay? So what we sometimes do is what we call recoding. And the goal of recoding is that the protein is staying completely the same. We're not changing the protein at all. But what we're doing is we're changing the DNA sequence that encodes that protein. And we're changing it so that we're using the preferred codons, the codons that get translated more quickly. So for those of you guys who are in iGEM, you might notice that sometimes when you go to the parts registry, you'll see codon optimized genes. I just saw one that was GFP codon optimized for plants, meaning that it's the same normal GFP gene, but the sequence that encodes it uses all the preferred codons of a plant, or something like that. And people will do that again to try to get a higher level of gene expression. Okay? So the beautiful thing about gene design is that it'll do this for you. So if we go back to the home page of gene design, and you now go to design a gene, and now you want to take the protein sequence, which I think is a little bit further down here. So proteins, again, I said there's 20 different amino acids. Each gets abbreviated by one letter. So each of these letters, there should be 20 different letters. Each letter represents one amino acid or one unit of the protein. Okay, so you copy that. You paste it into gene design. Okay, so enter your amino acid sequence. And then what you'll see down here is here's our 20 different amino acids.
And is it breaking those up still?
Yeah, I think that just has to do with the formatting of the web browser that it put in random spaces.
Oh.
I think you just broke it every 20 or something. Or every,
Doesn't look
the
right.
program will ignore the spaces. And what you'll see is we've got a bunch of different species here. So Saccharomyces cerevisiae has yeast, E. coli bacteria, humans, C. elegans are worms, fruit flies, and then the different bacterial species. And what it's showing you for each of the 20 amino acids is which of the codons is preferred. So in this example, lysine, AAA, is the preferred codon. Okay? But if you change the species, what you'll see is that those change. So in yeast, Saccharomyces cerevisiae, now AAG, actually happens to be the preferred codon in that species. So if I was moving a gene into yeast, I would change this AAA to an AAG, and it might speed up translation. Okay? And if you go through and you click the different ones, you'll see that some of them kind of change around. A gene, and let's say optimize it for humans, because I want humans to glow green. And sorry, I don't know why it's hidden back there, but you click the reverse translate, so it's going to take your protein and put it into a nucleotide sequence. And this is going to be the nucleotides using the preferred codons. Okay. So here's my nucleotide sequence, and then you can go directly into building block design. So it'll now take your human optimized GFP, and go back onto this screen, again use the defaults, and it'll give you the oligos for a human optimized GFP gene. If you go back, you can just switch the species. I can switch it to yeast. And reverse translate. And here you're going to get a different nucleotide sequence. You can tell right off the bat that it's different because the yeast sequence is 45% G's and C's. If you remember back, the human sequence is 63% G's and C's. Okay? So we started with the same protein, but we're winding up with a completely different nucleotide sequence because humans and yeast are using different nucleotide ladders, different preferences to encode the same protein.
So would it do the exact same thing, just some gene expression would be attenuated if you use the, like a suboptimal codon set?
Yes. Generally speaking, if you recode and use the preferred codon, there's a good chance to get a better gene expression level.
Okay. And can you manipulate that and request, I guess the algorithm set for?
Yep, I mean, this program will do it. Optimized her, I mean, this program will only do those 6 species for you.
But can you do it by hand? Is it
Sure.
you can just drop in an equivalent sequence that might be attenuated or might not be?
Right, what you need to know is for your species, which codons are the preferred. So what you need to know is this, right?
Right.
But once you know that, you can take your amino acid sequence and go through for each amino acid, put in the preferred codon, and design all of those for
So
whatever.
you put it on, preferred codon and preferred codon, so amino acids,
Exactly. Yeah.
reconcile.
So is it, the way I'm envisioning this, if you start like one letter over on the sequence, do you end up with, I guess I'm wondering about the triplets. Like, do they just start at the beginning and it's 1, 2, 3, that's an amino acid? Or if it
Yes.
started at the second,
Well, if you
or
start
it
at
only
the
breaks
second,
apart at the,
let's say this was G, G, G. If you
Yeah.
start at the second, here you're going to have an AAT, which coincidentally still encodes lysine.
right?
But then instead of having TCA, you're going to have a completely different one, CAG,
Right.
which is a completely different amino acid.
So my question is, how do you know where to start?
So a protein almost always starts with the amino acid ATG, which is methionine.
Okay.
So that's the first way that the cell knows where it started. It's looking for methionine, but it also needs sequence context. So it's looking for an ATG in the context of other nucleotides, which lines up the machinery that
Okay.
makes proteins at the right place.
So it does have a marker. Okay.
It does have a marker. That's what we call the RBS, the ribosome binding site somebody mentioned. The ribosome binding site is a sequence around the ATG, which sets you up at the start of a gene to say this is really the beginning where to start.
Okay. Cool.
So, sorry, start codons read as start codons regardless of frame. If you have an ATG occurring anywhere in the genome, does that register as?
No, so there's going to be ATGs in the middle of proteins, but because they don't have context, they're not read as start codons.
Okay. Context being
The
ribosome
RBS,
site.
the ribosome binding site. Other questions?
This drop-down menu, is that an order of preference or is it just?
Oh, yeah, I don't know for that drop-down menu. I think you can manually change it so that it's like the
Right.
Saccharomyces preferred codons,
So
with the
just the
exception
same.
of you can manually change 1 or 2 if you wanted to.
So you'd have to look at it.
Yeah. So this would be a way, I guess the program will do it if you wanted to do like a plant species, you could manually put in what each of the preferred codons was and then it would do it for you. How come yours? Oh, you're not automatic. I always wonder why that formatting is so weird. Okay, well, our reaction should be ready. Yeah. Oh, wow. Perfect. So, again, your negative control for there should go into a big tube and get labeled TPCR NC negative control. We'll leave it for next week. Your positive control from there goes into a separate tube. And then your actual assembly reaction gets diluted and put into the finished PCR you just set up. Any questions or confusions on that? Okay, ask me if you've got any questions or confusions on that. But you can come grab your sequences.
What do we do with the positive and negative control from here?
From here, so the 3 that you just set up for the finished PCR, your TPCR, your positive and negative, and your amplification finished PCR are all going to go into machine
Oh, okay.
to amplify.
Got it.
Yep.
Okay.
Then they're gonna run for another hour and a half, but the machine will hold them till tomorrow when Tom will come out and empty.